Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, Overcoming Challenges to Performing Single-Cell and Spatial Genomics Experiments. I'm incredibly pleased to welcome Dr. Ronan Chaligne. Dr. Chaligne leads the single-cell analytics innovation lab team intending to develop and deploy cutting-edge molecular profiling and data analysis solutions to support the broad community at Memorial Sloan Kettering. The sales team's mission is to assimilate emerging technologies in the rapidly advancing fields of single-cell sequencing and imaging-based profiling, in addition to providing robust analytical solutions. A very warm welcome to you, Dr. Chaligne. Following the presentation, we will have a Q&A session, and we welcome any questions you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I will hand over to Ronan to begin his presentation.
Hi, George. Thank you for the introduction, and thank you for giving me the opportunity to present today. It's always kind of a pleasure to present SAIL and my team works to people around that might be interested. And today I'll try to focus on what we kind of do every day. It's our bread and butter. It's kind of trying to improve single cell and spatial genomics experiment. Our team collaborating with a lot of different type of academic lab, can be translational research, can be more fundamental research. And we have been kind of facing different challenges and in sample preparation, in data quality and everything. So I'll try to kind of give you a general overview of what we've been through, and hopefully this will help you as well. And of course, if you have questions, you want to collaborate with us or have any question in the future, please don't hesitate to reach out. We are really here to help any lab that's facing challenges. I always like to start my presentation by just reminding everybody that even though we do single cell RNA-seq today, we all talk about this technology as a cutting edge method. It's that have been chased for many years, even decades, by researchers. And the first kind of publication showing an attempt to do that is actually early 90s, where you have this paper doing single cell RNA-seq. It's not really RNA-seq in the sense of we're doing today. They were kind of targeting one gene at a time. Obviously, there was no NGS at the time. And the way they try to quantify is not even qPCR, it's by doing thousand blot. But if we look at this paper, we can see that the overall method and logistic kind of is pretty close to what we're doing today. They do a reverse transcription started by a poly-DT. They amplify with PCR cycle, and then after they try to look at amplified product for here one given gene if it's present in the same amount in different cell. So it's something that people have always wanted to do, and we have kind of perfected it over the years. And now, obviously, we reach a sensitivity and a throughput that was not there in the early 90s. But I just like to remind myself that that's something we have kind of improved over the years, and the stage we are today is probably just one step along the path, where maybe in 5 years we'll have even more amazing throughput and methodology to capture information at single cell or spatial resolution. So the single cell era really exploded, I would say, with this paper from Azim Surani lab in 2009, where basically they did single cell more or less the same way it was done in the early 90s, but now they had NGS to read out the product they were amplifying at single cell level. And this is really the first paper that shows that you can do higher sensitivity at single cell level. Of course, obviously, today we do way more complicated method. NGS has been expanding over the years. And what my team is doing is, on a daily basis, is try to benchmark new method and equipment for single cell or spatial methodology. We also try to develop or implement new pipeline and new assay to answer like new biological question that are kind of asked by our collaborator in their model system. We try to optimize existing protocol. We try to also optimize analysis pipeline. I think there is as much novelty and innovation in the protocol as well as in the way people are looking at the data now, and they really try to squeeze and extract as much information as possible from those single cell methods. And also general things we do, it's, of course, as I said at the beginning of my talk, it's we guide, we advise collaborator or lab around us about their project. You can say that this is just our local experience, I would say, but number of samples we are processing every month keep increasing. So single cell is really becoming more and more popular. And now, since last year, spatial is even increasing this trend of people wanting to do more high resolution interrogation of their sample. And something we've seen also, this is a graph you can see on your right, is the number of library we're preparing per sample. And what this graph is showing is like in 2016, 2017, when we create cell, people tend to come with one sample and just do single cell RNA-seq. Now today, when people come, what they do is most of the time multi-omic approach, meaning that when you bring one sample, what we're gonna send for sequencing is not one library, but two different libraries, and those two libraries will have different content. So it can be people trying to do surface protein plus single cell RNA-seq. They can do chromatin plus single cell RNA-seq. So it's kind of trying to capture several layers of information at the same time. So I'm gonna go into the detail more after later in my talk. Something I also really like to start my talk with is the team that's doing the amazing work I'm presenting today, because I'm not really the one making all these things work and looks amazing. So I really want to have a big shout out to everybody in my team that's working on this. And also what I want to stress out is that we are kind of, there is more than 12 people in the team now, and half of them are experimental biologists working at the bench. Half of them are computational biologists working on processing the data, analyzing them. So, and I really believe that to make single cell or spatial technology the most efficient possible, you need those two type of skill set, like people that are really good at the bench, that know how to tweak or improve the method, but also you need people to know how to look at the data and how to improve kind of data analysis. So to get back to a more broader kind of a sense of what's happening in the field, this is kind of a schematic that have been made in 2021, but I think it's still true. This is all the information I think today, maybe there is more, but at least we can today capture with single cell. And if you look, like there is a lot of different information, it's going to be difficult to capture all of them at once, but definitely we enter an era where we try to capture more information together. And I put the mRNA in red because somehow mRNA is always the common information we are trying to capture in all this multi-omic approach. And I think there is several reasons for that. One is mRNA is definitely giving a good cellular state information about the sample we're interrogating. And that's why we try to capture messenger RNA first before anything else. The other reason, and that's why also I'm doing the work I'm doing, is it's because mRNA is technologically the most easy information to capture. And the reason for that is it's abundant in the cell. It's also showing a natural amplification of the signal. If you have a gene A that's not expressed, you won't capture any copy of mRNA, or like a handful of them. If a gene is highly expressed, you can capture thousands of those copies of mRNA. So the kind of change in dynamic between a gene not expressed and a gene expressed is going to be huge, and you can capture that efficiently in RNA-seq or single-cell RNA-seq. So there is this natural amplification of the signal. If you look, for example, now at chromatin accessibility, a gene that is heterochromatic or that's not expressed, you're not going to capture any copy of it doing attack. Now, if you have a gene that's highly expressed and you do attack, you're just going to capture 2 copies. So you move from 0 to 2 in the chromatin accessibility space if a gene is not expressed versus expressed, when in mRNA, you can move from 0 to 1,000. So again, mRNA is giving you this kind of natural amplification of the signal that you can more easily capture in single cell where data are sparse. And of course, mRNA is a nucleic acid molecule, so we can also PCR and amplify it way more easily than it would be if we try to capture protein or other type of information. So for all these reasons, I think that's why mRNA has been always the things we try to capture first. But today, and we talk about spatial, but for me, it's really trying to capture messenger RNA, gene expression, and we add on top of it some other information. And it can be surface protein, it can be lineage tracing, it can be DNA methylation, or it can be spatial information, where now we're going to locate in space where the molecules are, and then we can assign them to different cells. But in the end, for me, especially just another multi-omic approach where we've managed to kind of capture mRNA and add the spatial component to it. Of course, for all the reasons I just explained, single-cell RNA-seq is probably the most widely used single-cell approach today. There is a lot of different companies. There is a lot I've not even put on the slide there because I did not update it recently, but now there is Sparse Bio, there is CPO, there is Scale Bio, there is Fluent, there is Honeycomb, there is all this kind of technology company trying to develop kit with their specificity to capture a single-cell RNA-seq. Of course, the most widely used is 10x Genomics kit and methods. But I would say also, and that's why I put them side by side on the right, or like this paper did it, where they put the C1, which was one of the first commercial attempts to do single-cell RNA-seq. And you can see that the kind of information capture or the metrics or the throughput of those two methods is widely different. And what I like to say on this slide also is that even though 10x is most widely used and most preferred method probably in the academic world now, I feel that nothing is perfect, and you should really think when you start a project what method, what commercial platform might be the best fitting your need. It might be 10x, but there might also some other company which have other technology or platform that might be as useful or might answer better your question. So always start a project by thinking, okay, what's going to be the best approach that's going to help me answer the biological question. Next, I want just to dive a tiny bit into how to do single cell. And as I just said, the most widely used is 10x Genomics kit. I'm not going to spend too much time on this slide, but the point is the principle of 10x Genomics single cell RNA-seq is to kind of encapsulate in-drop single cell. And we call this like droplet-based a single cell RNA-seq. It has the advantages of being high throughput, being efficient, and also allowing like high sensitivity of capture of the RNA molecule. The way it's working is actually it changed recently. There is some other way, but the classical way to do it was to capture RNA using a poly A tail, and then you do reverse transcription. And at the end of the reverse transcription, you have a template switch. And for this to be efficient and capture with high sensitivity and high efficiency the molecule of RNA, what you want is healthy sample with healthy cell, and you want full-length mRNA. If your RNA is degraded, if your sample is suffering, you will have shorter RNA and you won't capture as many information or molecule doing single-cell RNA-seq. And what you also need is kind of, for that, is an intact plasmic membrane, so the cell being healthy. And also what you need is a low RNase activity. And when I say that, it's because based on what type of sample you're going to work, RNase activity in your sample going to be different. And a lot of the company or a lot of benchmarking are done on brain. And the brain is a good organ to do benchmarking because you have a really different cell type with wide range of cellular state. So it's kind of usually a beautiful data set you can obtain doing brain because you see all these different cells, the glial cell and the brain cell and everything. So it's kind of a really kind of a nice organ to study, I would say. But the other reason is that in brain, RNase intrinsic RNase activity is close to zero. And if you look, for example, at everything that's more internal organ type of sample, pancreas, spleen, lung, liver, colorectal, you see that RNase activity is definitely way higher just by the virtue of having kind of RNase activity within the organ. So when you're going to work with those organs, you know that there is higher chance your RNA get degraded, or at least your RNA going to be not as good quality. So I'm saying that because when people work on pancreas and look at brain data, they're like, oh, my pancreas data are not good. I would say that you have to set up expectation based on what you are working with. And it's difficult if you work with pancreas to expect high UMI capture, as you can see in brain. And you have to be kind of aware of those things and when you design your single cell experiment. The other thing that's pretty famous in single cell world in single cell, or I would say single nuclei attack-seq. And this allow you to kind of capture native chromatin structure, see what's achromatic, what heterochromatic in your sample at single cell resolution. But to achieve this, what you do is to have an efficient tagmentation, which is an enzymatic reaction done with a TN5. And to have this efficient tagmentation, you need to preserve the native chromatin structure, you need to treat the sample in a certain way, and you need to incubate your sample an hour at 37 degrees, which is when the tagmentation is going to happen. And the other thing you have to avoid is to have too much mitochondrial DNA in your sample. That's why most of the time ATAC is done on nuclei, because mitochondrial DNA will be tagmented as well and will give a lot of background on signal when you try to do genomic attack, a genomic DNA attack. And then this will kind of lower the quality of your chromatin accessibility single cell experiment. So why I'm saying this is because today we are moving more and more to multi-omic approach where we try to capture those different layers of information. And one of the really famous multi-omic approach is trying to do a single cell level attack plus RNA. And it's on the biology perspective, it's amazing because now you can link a gene expression level with a chromatin state. And as you know, as we know, probably epigenetic kind of regulation is something really complicated with different layer of regulation, and trying to understand and capture the gene expression activity with the chromatin state is something that people have been trying to achieve for a long time and giving a lot of insight in terms of cellular state, plasticity, all these kind of cellular mechanisms that can be important in kind of normal development, but also in disease. Now, on the technical side, because I am a technical person, it is challenging because I just showed in the previous slide, capturing those two layers of information require different treatment of your sample. And when we do multi-omic, what I'm really trying to say is that we have to compromise, we have to trade off. And there is no way if you do multi-omic approach, you're gonna get as good data as if you do standalone. And for example, in the case of doing single cell attack plus single cell RNA-seq, what you see is by doing the attack first, you have to incubate your sample at 37 degrees for an hour, and only after you can do the single cell RNA-seq part of the experiment. And there is no way to do it the other way around because we need the chromatin to be intact when we do the attack plus RNA experiment. So what I'm trying to say is that when we do multi-omic approach, attack plus RNA, by trying to get a good attack signal, we are gonna impact RNA quality because the first thing we do in the sample or in the protocol is to incubate the sample at 37 degrees for an hour. And when you work with sample where you have high RNase activity, like pancreas, colorectal, colon, lung, doing this step will somehow, even though there is a tiny bit RNase activity, will have an impact on your RNA quality. And that's why, for example, doing multi-omic approach on sample like internal organ will be more difficult than doing on something like a brain. So my point is just like when we do multi-omic approach, there is a trade-off to be done at the technical level, which will have an impact on the type of data you're gonna recover. And that's why I'm back to this main question. I'm always asking, what's your biological question you're trying to answer? Because based on this, you're gonna pick up what the best methodology you want to go. If you're really interested in understanding chromatin, yeah, maybe you should go for multi-omic. But if chromatin is just a secondary question and your main question is comparing maybe different cell type and look at gene expression, and that's what you do, then maybe you should not go for multi-omic because you know your RNA not going to be as good. Another thing I say frequently is like you need a good and efficient logistic. Again, for all these reasons, you need a good sample preparation and you need to preserve the biological information that's contained in your sample to be able to capture it efficiently. And that's why we also have worked on optimizing single cell isolation and tissue preparation for all these single cell and spatial methods. And to dive a tiny bit more into this, something that I'm, for example, always showing when we start a new project with collaborator is when you do single cell, there is two ways to do it. There is single cell or single nucleus you can do. And there is this paper from 2020, which is showing that if you do single cell or single nucleus, you're not gonna recover exactly the same cell type. And you can see on the left part of the slide that, for example, in single cell, you tend to not recover more fragile cell type. You're not gonna recover, for example, a macrophage at the high with high efficiency. You don't capture here it's a neuroblastoma. You don't capture neural crest cell or neuroendocrine cell, because single cell require you to dissociate the tissue, and the dissociation step is also something that's pretty harsh and damaging the fragile cell type, and that's why you don't see it in single cell. Now, when you do single nucleus, you don't do enzymatic dissociation. You snap freeze your tissue, you mechanically dissociate it. So this is a less harsh kind of approach, and it tends to preserve better the fragile cell type. In that case, with the nuclei, you can see you recover neuroendocrine, neural crest, you recover more macrophages as well. So, or fibroblasts, for example, or endothelial cell. And basically what nuclei is achieving is kind of preserving more fragile cell type. But on the other hand, if you look at nuclei, you see that all the B and T cell tend, or NK cell tend to be absent from the nuclei data set. And the reason for that is because immune cells have lower RNA content. And when you do single nuclei, you tend to capture less molecule per cell because you are not capturing the cytoplasmic RNA, which is not here. You're just capturing the RNA contained in the nucleus. And by doing so, all cells with lower RNA content might tend to be lost when you do single nuclei. Not lost because we don't capture that, but because there is lower RNA content and single cell is a sparse method, and we might not capture enough information from those lower RNA content cells. So I'm saying this to show, again, there is not a perfect method. Single cell can be amazing if you are an immunologist and really want to look at B and T cell. Now, if you want to really focus on more fragile cell type, endothelial, for example, single nucleus is most likely a better approach for you. So again, kind of the question you're asking really matter in kind of designing your experiment. And that's why we are working, and just one slide on this, trying to improve logistic. And the point of this slide is to show you that one approach can give you something if you are interested in that content, but if you are interested to something else, maybe there is other approach you should consider. Like the things we have optimized is different way to prepare tissue for single cell RNA-seq. So we have one way to do, which has been the gold standard, which is fresh suspension. When you have fresh suspension, you collect the tissue. Let's say it's a clinical sample. You collect the clinical piece of tissue from the surgery room, and then you have to dissociate, you have to fact sort, and then you have to do the single cell experiment. All these things require many people, a good logistic, a good coordination between different teams. The advantages of this is that you get single cell RNA-seq quality. So you get all the RNA from the cytoplasmic and the RNA. You can do site-seq, which is surface protein interrogation, because you get single cell suspension. You can look at surface protein. And you can also perform specific cell enrichment. If you, let's say, interested in only T cell, you can just look at T cell because you have fresh single cell suspension. You can enrich for those T cells. Now it's really challenging logistically, as I just say, and the enzymatic dissociation will have an impact on your fragile cell type, as I just showed in the previous slide. Another way we've tried to optimize has been to froze viably your sample, so which we call fresh frozen suspension. It's the same thing, except that we have a stopping point in the middle where we freeze with DMSO the single cell suspension. So you still have all the advantages of the single cell, prepare fresh, you can do RNA-seq, single cell, you can do surface protein interrogation, you can still enrich for some specific cell type. Now you still have to deal with enzymatic dissociation, and on top of it, you're adding another bottleneck in your kind of sample prep, which is freezing and thawing the sample. And we know some cell type will also be sensitive to this process of being frozen with DMSO and then thaw later. And this is adding maybe another kind of bottleneck where you're gonna maybe even counter-selected more of the fragile cell type. It makes it easier, you can collect more sample, process them more efficiently, but it might be not as good for the cell composition of your sample. So last thing is flash frozen tissue. So fresh frozen tissue. So fresh frozen tissue means you take the sample and you snap freeze it in liquid nitrogen. The big advantage is like logistically it's super easy. And you can collect as many samples as you want, and it requires almost no work and no money spent to kind of process them. And then we collect many samples and can decide later on what sample you want to proceed. So the logistic is easier. There is no real or big bias in the dissociation because we're gonna break the tissue mechanically, so we recover more different cell types than with single cell. Now, a big disadvantage of these things is that you cannot enrich for specific cell type, you cannot do surface protein interrogation, and you're just gonna capture RNA from the nucleus. So again, this is showing you there is no perfect method, but there is different methods that all have their pros and cons, and depending on what you're trying to achieve, this might have an impact on your conclusion at the end of your study, and what cell type you're going to recover and what information you're going to recover. Now, to try to go deeper in those sample preparation logistic, we have at Cell done kind of two comparison or benchmarking that I would like to focus a tiny bit now, where basically we try to assess method that people use regularly for their single cell prep and try to understand if there is bias also introduced in those preparations. It's been so standard and used for so many years, no one is questioning them, but we're like, okay, what's going to be the impact on using one or the other method? And the first one is trying to understand when you select for live cell with different kits, is this impacting the cell type you're going to recover or the data quality? So for that, we have compared 5 different methods which are pretty widely used. The first one is, of course, the flow cytometry, where we select for live cell by using DAPI or 7AD. Second method is Levitas bio-mash instrument called LeviCell, which is basically selecting live cell by selecting the cell by their physical integrity and if the cell membrane is intact, by making them levitate. So basically, they apply a force field, and the cells that are live will float at the top. Every debris or dead cell will just sink at the bottom, and then you can flush the channel and then recover the live cell at the top. And it's really gentle. The pressure inside the Levy-cell is way below what you can get in the flow cytometry. So that's why we pick up this method. And we also pick up 3 other methods, which are column-based, and they are all kind of, technically speaking, tiny different way to select for live cell, but they're all based on annexin 5. But some are going to be based on microbubble, some are based on immunomagnetic selection. So that's why we wanted to try to test those different methods and see what the impact on the sample quality. We select 2 types of sample, lymph node and CSF, cerebral spinal fluid. And we select those 2 types of sample because we don't need to dissociate them. They are kind of already almost single cell. Lymph node, you just have to down homogenize it and you get single cell with few minutes without really enzymatic dissociation. And cerebral spinal fluid is something where you already have single cell suspension. So we don't have to apply a single cell dissociation step, which would most likely damage and already change the cellular profile, the transcriptional profile of the cell. And we know that both samples contain fragile cell populations. So we wanted to see if those methods will impact the recovery of those fragile cell types. So this has been done in collaboration with 2 amazing collaborators at MSK, Chrissy Brown lab and Adrienne Boer lab for each of the samples. So thanks for them for helping us doing this benchmarking. So as you can see here, we compare the 5 methods in terms of viability post-selection, and we can see that there is no huge difference. Total yield might fluctuate a tiny bit. FACS is always the one with a lower yield, but there is no huge differences, and I don't think any method is particularly bad at selecting live cells. So for this, I feel we are pretty on a kind of similar efficiency, and the method you choose won't, in theory, impact really how much live cell you recover after. Now we did selection of those 2 types of sample, and then we performed single cell RNA-seq. And this is a traditional matrix we're looking at. There is a lot on this slide, and I'm sorry for that, but the point of this is really showing that all methods show good quality data at the end. There is no method where we see like abnormal high ambient RNA, method where we see lower cell recover or lower UMI count per cell recover. They're all kind of performing the same. There is some fluctuation, but it's n equal 1 for each method. So I don't think it's really something we should be worried about or like that's really significant. So in that perspective, this is pretty similar across the board. The thing we look next is what's really matter for us in this benchmarking is how much each cell type we are recovering. And this is something that's done in my team by Toby Crows, and that did an amazing job at cell typing the different population we are recovering. So this is an integrating UMAP of the 5 methods, and you can see on the right like all the different cell type we are recovering. And if you try to kind of overlay what method is contributing to what cell type, you can see there is some differences. And the biggest difference appear on the bottom left cluster, where you can see there is a kind of big difference between the different methods. And this is also highlighted with a red square on the table. And what you can see is basically the cancer cell, because this is a CSF. Sorry, I should have said it's a mouse model in which we have injected cancer cell, and the cancer cell migrate to the CSF, which is a cerebrospinal fluid. So what we are expected to recover here, it's a bunch of immune cell type and also cancer cell. And what you can see is here, if we use FACS, we barely don't recover cancer cell. And one of the reasons is that metastatic cells going into CSF are really fragile. CSF is kind of a really specific environment with specific kind of nutrient there. And the cancer cell in this CSF space are really fragile. And we know that when we get them out of the CSF and kind of prepare them for single cell method or others, they tend to die quickly. And what you see here is like FACS will barely recover any cancer cell, when other method with Levy-cell ahead of them is recovering about 10 to 8% of the cancer cell. So this is showing you that FACS definitely a harsher method, and all other methods are doing a better job at trying to preserving fragile cell type. What you can see also is the same for fibroblasts, where FACS is recovering, even though it's a small population here, fibroblasts tend to be not recovered by FACS, but they are by other method. But on the other hand, what you see is that we seen a lot of unknown cell type in the different method that's not present in FACS. And I did not have really time to put this on this slide here, but what we have identified as a known cell type here is mostly apoptotic cell, which we cannot basically attribute to any specific cellular state or cell type because they are being apoptotic. And what we see is that Levy cell tend to recover more apoptotic cell. Other methods that are more gentle recover also about 10 to 15% apoptotic cell. On FACS, you don't capture anything. And this is, again, I think, going into the direction of showing that FACS is a harsher method, but also selecting more efficiently or harshly, depend what your question, again, for live cell. And other methods tend to be more gentle. So again, I don't think FACS is bad and the other are better. I just think it depends on what the question you're asking and what you want to kind of look at. But definitely there is some differences between those methods when you select for live cell. The second sample we've been doing this is a lymph node, where we did the same exercise, cell type annotation, and then looking at what the contribution of each method for the cell type we are recovering. And here we see the same kind of thing, which is maybe not as obvious as the previous CSF sample, but with lymph node, we can see that, for example, macrophages, which tend to be more fragile cell type, or dendritic cell, tend to be less recovered by FACS, even though we can see differences in dendritic cell for Miltenyi, for example, or for macrophage, where it also as low as a FACS. So one that really stand out in those fragile cell type is Levy cell, which really seems to be the most gentle approach, where you recover the highest of those fragile cell type. So again, the point is, conclusion here that FACS seems to be harsher, and other methods seems to be more gentle. But again, if you are really interested here in B cell, you don't really mind having to use FACS to select those B cells. If you're interested in more fragile cell type, maybe FACS is not always the best method to select for live cell. So in conclusion, what we can say is that FACS seems to be harsher, but it's also, I did not show that on the slide, but it's also showing a tiny bit less ambient RNA and a clear distinction between anti-drop and drop with a cell in it. Now, Levy cell on the other end seems to be capturing more efficiently fragile cell type and also maintaining most apoptotic cell within the live cell pool. So it can be something you don't want, but also something you might want. So again, completely dependent of the question you're trying to ask. The second benchmarking we've done is comparing nucleus isolation, because I've said nucleus is inducing less bias in cell type recovery. Apparently, we recover more fragile cell type, but we wanted to compare because there is, over the past 2 years, a lot of new commercial kits to perform nucleus isolation from frozen tissue. What we compare is 5 different methods, 2 automatic commercial, GentleMax from Miltenyi and Singulator from S2 Genomics. We compare also the nuclei isolation kit from 10x Genomics, which is manual with a dense homogenization done in a tube, and also the 2 methods that were published in the method I show at the beginning of my talk, where they use manual dissociation and use 2 types of buffer to recover cell type. On the right graph, on the right screen, is a graph where my team have been kind of assess what the throughput, but also the star is how much they like to do each method. And you can see that this automatic method tend to be, of course, the most easy and convenient one to use. And the manual one, like an STTST or even 10x Genomics, tend to be not as fun because you really have to be doing it manually. And it's also adding some variability between samples when you do it manually and not automatically. So here we have used 3 different samples: glioblastoma, which is brain tumor. So it's more in the range of low RNase activity, because here RNase activity has a big impact on the RNA-seq signal we're going to get after. We've done colorectal, which is more around lung or spleen in terms of RNase activity. And we've done liver, which is in the middle of those two other samples in terms of RNase activity. All those samples have been done with collaborator, glioblastoma with Kenny Yu and the Tabar lab. Colorectal sample have been obtained with the Karuna Ganesh lab, and liver sample from healthy mouse has been obtained from Scott Lowe lab. So I really thank our collaborator for helping us with that. So we've done the same exercise, which is looking at different cell type we recover. And for example, when we did the glioblastoma, we can see that there is no big difference in the different cell type, and we tend to recover similar cell type no matter what method we use. I'm not showing on the slide, but automatic method, Singulator, Miltenyi, which also tend to be faster in extracting nuclei, tend to have the highest UMI capture. But overall, we did not see like big differences. The second sample we compare was colorectal, which has higher RNAs. We did not include Miltenyi because we forgot to put RNase inhibitor. And if I go back in this slide, you can see that Miltenyi in colorectal is lower in terms of UMI recover. And mostly this is because we did not put RNase inhibitor. So it was a good kind of positive control that RNase inhibitor is really helping protect the RNA. But if you compare the other method, you can see that there is no big difference in the cell type recover, but you can see that there might be some difference in the UMI you are recovering in the colorectal sample. Now for the last sample we've done, which is healthy mouse liver, you can see that this one tend to show more differences in the cell type recover, and the three commercial platform tend to recover the most diversity, and Singulator and Miltenyi tend to be recovering even more of the kind of fragile and unique cell type that, for example, 10x or NSTR-TST tend to have more difficulties to capture. But the thing is, we don't capture in that case here immune compartment in NST and TST. But we believe that this is again due in those cases that lower UMI done on nuclear RNA-seq has an impact first on immune cell type because those have lower RNA content. So we tend to have more difficulties to recover. And since Singulator and Miltenyi here are doing a better job at capturing efficiently higher molecule of RNA in nuclei, it tends that here we are capturing some B cell, even though it's not a big proportion of them. We're capturing B cell and we're capturing NK cell, which we are not seeing in NSTR-TST. So as a general kind of conclusion, we can say that Singulator and Miltenyi are performing better overall. There is different variation in cell type, more than what we've seen when we did the live cell enrichment. And that's why you probably have to be mindful when you do nuclear RNA-seq what method you want to go with and what is available. And I would say that our personal preference here at Cell is kind of automatic, reproducible nuclei preparation will make something more consistent over the different sample you might process and also give you probably higher quality data. The next thing I want to show is that all this benchmarking we've done have been based on the fact that we want to do single RNA-seq by capturing RNA using a poly-DT. But recently, like last year, 10x Genomics has been releasing this kit where now you can capture RNA-seq by using probe. And the big advantage of this is that now you can capture RNA that are fragmented. So even if your RNA is not full length, you can capture the small part of the RNA by using this flex kit, which is based on probe of 2 × 25 base pair. So even if you have a molecule of RNA, which is like only 100 base pair, you're going to still capture it efficiently. And the big advantage of this kit is that now you can start accessing samples that were not accessible before. And one of the big type of sample we have in every biobank in the world is FFP. So it's fixed formaldehyde paraffin embedded sample, where you take clinical sample, you fix it, or like a mouse sample, you fix it, and then you can store it forever. The caveat of that is like fixation with formaldehyde will fragment your nucleic acid. So RNA, DNA going to be fragmented. And if you use the sample for classical poly-DT approach for RNA-seq, you're not going to capture efficiently RNA. But with this kit, now you can access those type of samples. And this is a paper published by Luciano Martellotto group, which is showing the first how to basically extract nuclei from those FFPE efficiently, and then apply it to flex to capture efficient RNA-seq. We have also kind of worked on trying to make this more streamlined, and we have worked with S2 Genomic company to automate the nuclei extraction of FFPE. And this is, again, a collaboration with Kenny Wu, where we basically are using an FFPE from a glioblastoma sample, and we have kind of perform flex on this sample. And the amazing things we see here is that we capture an amazing good data set with like more than 6,000 UMI per nuclei on average, which is really good for a single cell RNA-seq data set. And what we've seen also is that we are capturing cell type that we usually don't see in single cell RNA-seq. So again, this is testifying of how your sample prep might have an impact on the type of cell, on the quality of the data you are recovering. Some of the things that you need to keep in mind when you perform single cell RNA-seq is the capture rate. People tend to think that if you have 10,000 cells in your tube, you're going to capture 10,000 cells in single cell. Like every method will have an inefficiency in capturing, and 10x is around 2/3, 66, 65%. If you do, for example, plate-based approach, you're not just going to capture 25% because sorting cell in one plate, in one well per well in a plate, is not really an efficient method to capture cell, and you tend to lose 3/4 of the cell by doing so. You need also to be mindful of what the biggest population of interest you want to interrogate. Is it a small population, a big population? And this will inform you how much single cell you should capture. You should also be mindful of how many samples. You can now multiplex sample easily with hashtag or other method. And you should also be mindful of the quality of your sample. Like if you bring a bad quality to us, there is a low chance you're going to get good data out of it. There is even cleaning up the data and everything won't help. Like high viability cell, high quality nuclei, all these things will help you get better data at the end. As I was mentioning, 10x is providing a lot of different approach now for multiomic and other company too. But there is also a lot of lab that are kind of trying to access even more single cell information. And I just want to spend like 2 slides kind of highlighting those methods. For example, one that we have implemented cell efficiently is trying to capture mitochondrial RNA or DNA to try to infer lineage tracing. And this is something I have on one of my first slides, which is showing how you can, using single cell approach, to extract even more information to kind of answer the biological question you have. Another approach we have kind of efficiently implemented cell is dogma-seq, which is multiome approach by 10x. But in this case, you can also add mitochondrial DNA into the mix, and you can also add surface protein information into the mix. So this is kind of a single cell approach where you will have 4 layers of information coming from the same sample. And this, I feel it's kind of amazing how the single cell field has been moving over the past few years and how much now you can capture information. And this is helping tremendously researchers to kind of answer new biological question and get more insight into their model system. Now, the latest advance, in my opinion, of all this multiome approach is spatial. Because spatial, as I say, just capturing molecule of RNA, but instead of doing it in single cell, you can map not molecule to one specific cell with a cell barcode or other method. Now you're going to map this molecule to a spatial coordinate. So, but at the end of the day, it's roughly the same kind of thinking behind it, just that the extra information you're capturing is different. The method we have used and we have now at Cell is a Xenium by 10x Genomics, which is providing in situ kind of approach where you can now map precisely within the cell what transcripts are present. The data set I really want to present in the last 5 minutes is this data set we've generated again with the Adrien Boer lab, which is one of the amazing collaborators we have, where we've looked at the same sample I showed earlier, where we look at a mouse brain that have been injected with tumor cell, and you see you can have like metastatic cells. So we've done a couple of different cell line, cancer cell line injected in the mouse, and we have kind of sacrifice the mice, take the brain, embed the brain in formaldehyde after fixation. So we did an FFP, and then we kind of apply those different slices onto Xenium slide. And we've done 4 FFP block with 3 brain slices each. And on the right, you can see the matrix of number of UMI recovered and number of unique gene recovered based on the different region and based on the different model system we have used. And what we've been pretty happy about is that you get something consistent in terms of number of cell recovered, number of the region we are looking at in terms of number of UMI or unique gene we're capturing, we see consistency across the board and across the different sample, which was something giving us confidence. Now, just to, in the last few minutes, to kind of tease out what kind of extra information as this spatial method can give us, we are just focused on the melanoma sample. Here on the right, you see the healthy control with the gene clustering by color, where you see all the different type of cell you have in the brain in LC. And you can see in melanoma that it's roughly the same things. You just have some metastatic cell in the tumor, in the CSF compartment of the brain, which is this kind of around the brain fluids that preserving the brain from being damaged when there is shock. And you can see that using a tumor cell marker, we can see where those tumor cells are. And we can see there is some even like infiltrating in the brain. And then if you look at the clustering on the right, this clustering is done by cell type, but also by the proximity of those different cell types. And you can see that the tumor brain are pretty far away from everything else in the brain. And you can see they're forming metastatic at the bottom of the brain in this specific tissue. Now, if we kind of try to understand better what are the cells that might be interacting with those tumor cells. And we see this kind of cluster number 32, which is the nearest tumor cell cluster, the nearest normal cell cluster to the tumor cell. So we have kind of dived a tiny bit into this cluster to try to understand what it was. And you can see that this cluster is really located right where the tumor cells are, also on the spatial map. And this cluster is really a small fraction of the total of the cell we are seeing, but it seems to be specifically interacting with the tumor cell. And when we have looked at what this cluster can be, and this again, it's kind of really quick overview of what we have done, we can see that this cluster actually is kind of identified with a cluster with a marker, which is a marker for the macrophages, CD53. And the interesting thing is that if you look at cluster 32, it's like the closest to 6, which is a tumor cluster. But if you look at 13, cluster 13, which is just after 32, those are also macrophages. So what we've seen, just looking really quickly into this kind of spatial data set, which was not something we could have found doing single cell before, is that we have a specific subcluster of macrophages, which tend to be identified as macrophages, but which tend to be really close to the tumor cell. And then if we dive a tiny bit more into what are those macrophages, we find that those are like activated macrophages, and they tend to be the one interacting the most with tumor cells, most likely because they are trying to either kill or eat those tumor cells and have some interaction with them, which is something we would never have seen or not as clearly or understand as clearly in a single cell data set. We might have probably see some macrophage are activated in our data set, but doing spatial now, we can tell that those are the ones directly interacting with the tumor cell. So this was really a quick overview of the spatial method, but that's the kind of approach now we can do and information we can answer. And one of the good things also we can do now with spatial is that as for single cells, there is different type of spatial, and there is, for example, a Visium from 10x, which is not single cell resolution, but will give you a full transcriptome from the spatial. So with Visium, you can get like tiny kind of tiny bulk of cell. You can have 5-10 cells clustered together under one spot. So it's still spatial, but the information you're going to get at the transcriptome level is an average of 5 to 10 cells. Advantage of Visium is that you get full transcriptome, but you don't get single cell resolution. So what I'm trying to say here is that you basically now have different type of method that can help you answer different type of question. And for example, here, which is a bioarchive published by 10x, what they're showing and what I really like about this slide is it's showing you that, for example, if you do Xenium, you can capture a lot of cells. Here it's about 150,000 cells, and you can get a single cell resolution. However, number of genes you can interrogate doing this method is lower than if you do a full transcriptome approach like Visium, where you can capture way more genes. For example, here you're capturing more than 5,000 genes per spot, but the number of cell you're capturing is way lower, and you are not even capturing single cell. So again, I just want to conclude saying there is different method, whether it's single cell, whether it's spatial, that can be useful to answer your biological question. But the first really thing you have to think before starting a project is what I'm trying to answer, what are the questions I'm asking, and this will tell you what method is the best fit for your needs. I really want to thank everybody in my team that have been helping with all the benchmarking and doing this amazing job of implementing new method, testing new pipeline, developing new method. I also, of course, want to thank all the collaborators we have at MSK, without who we could not do anything of this work. And of course, all the clinicians and all the patients and their family for being so kind to provide us with sample and help us understand better disease and how to kind of fight them. So thank you for listening to me today, and I'll be more than happy to take any question now.
Thank you very much, Ronan. That was a fantastic presentation. And as with all the other talks, we're likely going to have lots of questions from the audience. And I can see that we have received some already, so I'll move straight on to the Q&A now. So your first question is, do you think single-cell methods will tend to not be used in the future because of the development of more performance spatial technologies?
So I think spatial will definitely kind of use some of the single-cell field, I would say, or like resources in the research environment, meaning that people that were doing single cell might now instead do spatial. Now I don't think single cell will completely disappear, because there is many things I show in my talk that single cell can only achieve, like a lot of the multiomic approach where we capture chromatin state, where we capture maybe mitochondrial lineage tracing, are only single cell available right now. And another thing I think single cell is allowing that more and more people are doing, it's functional screening, like all that CRISPR screen, like lineage tracing, all these kind of things are done in single cell, because you have a cell line, a model system where you try to apply some perturbation or like some screening, and then the throughput or the readout of this kind of method is single cell, and it will be not really kind of something you can do in spatial technology. So the single cell field might switch, but I still believe single cell is offering a lot of opportunity or access to some biological information that spatial is not able to offer.
That's excellent. Thank you so much, Ronan. And moving on to your next question today, what is the most frequent use case for in situ spatial methods currently in your team?
So I think this is more kind of related to the previous question, where basically, again, spatial will be people that tend to benefit from spatial the most because of the samples they are trying to look at. And most obvious case is people working in the cancer or disease context, because you have tissue, and as I show, like dissociating a tissue will have an impact on the cell type you recover and the information you're getting. So especially tissue where you have high RNase content, like pancreas, colon, all these things, if you take a piece of tissue and you have FFPE done, it's preserving the tissue quality, preserving the cell type, because it's fixed immediately, nothing can happen to it. And then spatial will kind of give you a lot of new information we did not add with single cell approach. So I think those specific contexts, which are clinical contexts, are really useful for spatial. The other type of people is people working more in mouse model or in development, and want to understand how cells are migrating, how cells are kind of interacting with each other, and that's where we see most of the in situ spatial use right now.
That's fantastic. Thank you, Ronan. We have time for perhaps a few more questions today. So moving on, do you think spatial biology will help uncover new biology?
Totally. I think it's something we our people have been expecting for a long time, to be able to do such a high throughput in terms of number of cells, but also in terms of number of genes in spatial. And by just a small example I show at the end, with the collaboration we have done with Adrian Boer laboratory, you can see that we are already immediately picking up on like something that might seem super obvious when we look at the spatial information, but like single cell was not able to provide us this type of information. And I think also spatial will give us maybe cell type we were losing when we were doing single nuclear, single cell because of they were fragile or they were too small in proportion. Like the activated macrophages represent 0.5%. So when you do a 10,000 cell single cell experiment, 0.5% is really a low amount of cell. It's going to be like, if my math are correct, it's going to be somewhere 50 cell you're capturing when you are doing 10,000 cell. So it's really no, my math are wrong. It's 500 cell. But anyway, it's going to be like a small proportion of cell, and it has to be also macrophages, which is fragile cell type. So you might lose them during the single cell dissociation or during the preparation of the sample. So yeah, I feel spatial will maybe hopefully give us like a more, I would say, true idea of what cell type we have in samples and how they interact with each other.
Thank you so much, Ronan. And we have time for one final question today, which is, do you think high resolution methods like Visium HD will make in situ methods less relevant?
I think the question will be how much sensitive Visium HD is. So Visium HD is this method where, as I show at the end, with Visium you can capture full transcriptome, but you cannot capture efficiently single cell because it's like the spots are like on the slide are pretty big, and then what you capture is just an area where you have few cells. So with smaller spot and higher resolution, we hope we're gonna achieve single cell resolution. But yeah, the question is how efficient is gonna be the capture of the gene, and I think comparing, for example, in situ method with Visium HD will, I guess, in the future tell us what method people might prefer. But there is a chance that yeah, Visium HD gonna be one, if it's really sensitive, one of the methods where people will be more willing to do because they're gonna get even more information out of it than in situ. But I guess maybe different sample with again different requirements or different topological question asked will benefit from one or the other method.
Thank you so much, Ronan. Unfortunately, that is all that we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thank you once again, Dr. Chaligne. It's been an absolute pleasure having you present with us. And for everyone watching, we'll be back tomorrow at 8 a.m. Pacific for another fantastic lineup of talks. Thank you and goodbye.
Thank you, George.